Hello, welcome to Breakthroughs from Science Mission. I'm Sadashiva Pai and going to discuss a couple of breakthrough papers published in the week of July 14. Let's start. First, we'll discuss a paper published in Nature on the role of recombinant FGF1 in treating diabetes. Why is this paper so important? Role of FGF1 in lowering blood glucose levels is not clearly understood. Authors demonstrate acute and chronic FGF1 treatment lowers glucose levels in different mouse models. Authors also show that recombinant FGF1 lowers glucose levels probably through insulin desensitization. Let's look at the data. A single injection of FGF1 was sufficient to attain normoglycemia in severely hyperglycemic, genetically obese mice, and the effect was dose dependent. Glucose lowering effect was also observed in another genetic model and diet induced obese mouse model. FGF1 had no effect on blood glucose levels in normoglycemic chow fed mice, but reduced glucose levels under fasting conditions, thereby dissociating the glucose lowering effects of FGF1 from its effect on food intake. Also, FGF1 seems to be unique in its ability to lower blood glucose levels compared to other family members, indicating specificity of FGF1. Alternate day injection of FGF1 for 36 days also lowered glucose levels, while the body weight did not change. FGF1 treated mice showed a marked improvement in insulin sensitivity as measured by insulin levels and insulin tolerance test. Triglyceride levels were decreased while glycogen content was increased in livers after 5 weeks of chronic treatment of FGF1. How does FGF1 reduce glucose levels? In streptozotocin, that is STZ-induced diabetic mouse, wherein drug caused destruction of insulin-producing beta cells, FGF1 alone failed to lower blood glucose levels, indicating that FGF1 is not insulin mimetic. Also, pretreatment of STZ mice with FGF1 markedly enhanced the glucose-lowering effects of exogenously supplied insulin. The FGF1-dependent increase in metabolic clearance rate was severely blunted when insulin secretion was inhibited by somatostatin. To discount the mitogenic effect of FGF1, proliferative activity of truncated FGF1 showed decreased glucose levels similar to untruncated FGF1. Similar results were also seen in diet-induced obese mouse. FGF1 receptor knockout mice failed to lower blood glucose levels, indicating the action via the receptors. These results conclusively prove a role for FGF1 in lowering blood glucose levels. Next, let's look into a paper published by Oded Rechavi and colleagues in Cell. Authors demonstrate that starvation leads to inheritance of small RNAs for 3 generations. Why is this paper so important? Starvation health effect on future generations and mechanism of inheritance is not clearly understood. The study shows the inheritance of small RNAs for 3 consecutive generations following starvation. Authors identify genes targeted by transmitted small RNAs. F3 offspring also show an increased lifespan. Authors starved C. elegans for 6 days and looked at the small mRNAs. This figure shows the identification of STGs, that is, small RNAs targeting specific genes in different groups. More than 500 predominant up and down regulated small RNAs are 22G RNAs, and out of those, 88 were downregulated mRNAs. 22G STGs that were upregulated in their ancestors following L1 starvation were also upregulated in statistically significant manner in the F3 progeny in comparison to the fed ancestors. Potential target gene enriched were found to be related to nutrient reservoir activity. In addition, small RNAs may indeed preferentially target germline expressed genes, which are 27% up and 8% downregulated. Authors also found that inheritance of 93% STGs is dependent on RDE4 as shown in this figure. Authors also found that offspring third generation from starved great-grandparents show extended lifespan in comparison to the ones that were continuously fed over generations. Authors came up with a model that suggests that a transient trigger L1 starvation is memorized by the generation of an ensemble of small RNAs, which are then available to target mRNA in ensuing generations in the absence of the original starvation trigger. Thank you for watching Science Breakthroughs from Science Mission.